Hello guys and welcome back to Accumulation Academy. So this is lecture third for glycolysis, and till now we have discussed about the different 10 steps which are involved in glycolysis, and we have also mentioned like what are those three irreversible reactions which is important and the determining step in the glycolysis, right? Now further we are going to discuss about the phase of the pyruvate. Okay. So energy-rich functional groups in the substrate of glycolysis. So what does it mean? This energy-rich functional groups, which is going to give you more amount. So these energy-rich functional groups, which like on breaking down the bond, is going to give you more amount of energy. Okay. So in the case of glycolysis, what are those? The enol phosphate in the case of phosphoenol pyruvate, carboxyphosphate in the 1,3-bis phosphoglycerate, and the thioester bond, which is present in the active site of glyceraldehyde 3 hydrogenase. ठीक है? तो ये portion ऐसे है which is going to give you more amount of energy. Okay, so now we are going to discuss about the fates of the pyruvate. So pyruvate can be either वो ester CoA बनेगा जो Krebs cycle में enter करेगा and it will give you another huge amount of energy, or it can become lactate or ethanol, oxaloacetate or alanine. Okay, so depending on the conditions, aerobic, anaerobic conditions, we are going to talk now about the fate of pyruvate. So let's talk about first the anaerobic fate of pyruvate. That is fermentation. So the reduction of pyruvate can occur in two ways: homolactic and the alcoholic fermentation. So what is homolactic? So नाम से पता चल रहा है, it will result in the formation of the lactate. Now in our body, like for example, can you tell me when there is less amount of oxygen supply, the lactic acid gets accumulated. Okay, now that causes pain in the body. Can you give me one such example? That is low amount of oxygen and lactic acid deposition in our body, so which gives pain sensation also. One example, such example, can you give me guys? Yes,
during exercises.
Okay, it's little correct, but again, be more specific. Kab? Which kind of exercise will give you lactic acid accumulation? Yes, Stanley? More? Give more thoughts. The answers, everything you know, I'm sure.
Ma'am, can you repeat the question?
So there will be a low amount of oxygen, okay, and in the body there will be a lactic acid accumulation. Of course, there is a less oxygen. That's why it is undergoing anaerobic fate of pyruvate. Forms the lactic acid in the body, human body. I'm talking about. Okay, so this lactic acid accumulation will give pain in the body. So when does this happen with us?
Ma'am, when we do heavy exercises?
No, actually. See, the thing is, low oxygen condition comes when you actually run fast, right? So, you know, we take deep breaths and all. When we are short of the breath, like we are running sprints, like those guys who do 100, 200 meter race, all of a sudden in fraction of seconds they are completing, like Usain Bolt, right? All these guys. So what happens that time? अगर मैं अभी अचानक से 200 meter दौड़ूंगी, like to my maximum fastest speed, what will happen? My knees will start paining, right? And after some time, when my breathing becomes normal, the pain will also go. So that is lactic acid accumulation in my joints, where this because of the low oxygen level, it is getting accumulated, and this accumulation results in the pain in my body. अभी समझ में आया? Right, because I'm making
Yes,
my muscles and skeleton go so fast, and मैंने इनको time भी नहीं दिया, oxygen नहीं दे रही हूँ. So I need certain amount of I really need very fast requirement of my ATP because मुझे उतना दौड़ना है. तो मुझे कैसे मिलेगा? I can't wait glycolysis to complete the Krebs cycle and then make ETC, and there is no oxygen also, right? So ETC happens when the oxygen is your electron carrier, right? So suppose oxygen is not there, so all these processes not able to get complete. तो वहाँ पे क्या होगा? Then this will result in the lactic acid formation, जहाँ पे आपका ऑक्सीजन कम रहेगा. Okay. So homolactic and alcoholic fermentation which occurs in the muscles and yeast. एक example दिया, homolactic हो गया आपका muscles में and alcoholic fermentation हो गया yeast में. So respectively depends on the reduction pathway of NAD+. तो ये किस पे depend है आपका NAD+ में. So when we are using NAD+, so what kind of enzymes you are going to use? Dehydrogenase, right? I have mentioned you many times. जहाँ भी आपका dehydrogenase रहेगा वहाँ पे आपका NAD+ रहेगा. So if you talk about lactic acid, then what will be the name of the enzyme? It is going to be lactate dehydrogenase, right? LDH. Similarly, for alcoholic fermentation, the name of the enzyme may be then simple. Alcohol dehydrogenase. है ना? Simple हो गया? Okay. The NAD+ formed during glycolysis should be reduced in NADH and GAPDH. So in the presence of oxygen, the reducing equivalents of NADH pass into the mitochondria for reoxidation. So glycolysis में जो भी आपका NAD+ बनता है, वो आपका mitochondria में चला जाता है for the Crabtree cycle. Okay. Now suppose it is under anaerobic condition, then it is reduced in the above mentioned form, यानी कि in the lactate formation and in the alcohol formation. Guys, this much is clear to you?
Yes,
Okay.
ma'am.
Yes, ma'am.
Okay, now look at this figure. So glucose glycolysis results in the formation of your NAD+. अभी pyruvate है हमारे पास, ठीक है? So pyruvate जो आपका NADH है, this is what we are going to use it, reoxidize. अभी यहाँ पे क्या हुआ है? Reduce हुआ है. What do you mean by reduce? Addition of H+, है ना? अभी हमें यहाँ पे use करना है, so it is going to be oxidized, reoxidized. So pyruvate to lactate, homolactic fermentation, आपने NADH use कर लिया है, NAD+ मिलेगा, ठीक है? यहाँ पे alcoholic fermentation, NADH again use कर लिया, you are going to get ethanol. अगर अभी citric acid cycle होता है, then also वहाँ पे you will get more amount of NADH. Now NADH फिर ये जो है आपका use होता है आपका ETC, ठीक है? So this is just the overview, like what all fate the pyruvate can have. Now let's talk about homolactic fermentation and lactate formation in detail. Simple है, कुछ भी नहीं है यहाँ पे. You can see glucose homolactic अगर जा रहा है, so of course glucose 6-phosphate to fructose 6-phosphate, fructose 1,6 diphosphate or bisphosphate. Then you have got the glyceraldehyde 3-phosphate, dihydroxyacetone phosphate, then 2 pyruvate molecules, and this will result in the formation of 2 lactate. ठीक है? अगर हेट्रोलैक्टिक होता है, ओके, सो हेट्रोलैक्टिक में क्या होगा? सेम थिंग, इट्स जस्ट दैट glucose 6-phosphate will be converted into 6-phosphogluconate. Okay, what is happening over here? Glucose 6-phosphate is getting converted to 6-phosphogluconate, ribulose 5-phosphate. Then it is giving you. So guys, यहाँ पे देखो क्या हुआ है? This is 6 carbon, this is 6 carbon. And ribulose 5p is actually your 5 carbon, है ना? तो यहाँ पे क्या हुआ है? CO2 has been released out. अब क्या होगा? Glyceraldehyde 3-phosphate is 3 carbon, acetyl is your 2 carbon. Then pyruvate, अगर pyruvate है 1 molecule, so you can get lactate, and then from here acetaldehyde ethanol. So 1 molecule of lactate and 1 molecule of ethanol. Whereas if it undergoes homolactic, you are going to get 2 molecules of lactate. Yeah. So this is a feature of muscles occur particularly during vigorous muscle contraction. In rapidly contracting muscle cells, the demand for energy is high. Now, when the oxygen supply is depleted, lactic acid fermentation provides sufficient NAD+ to allow glycolysis to continue for a short period of time. ठीक है? तो अभी क्या होता है? See, guys, like when you are running, okay, when you're contracting your muscles so fast and you don't have sufficient oxygen, so it is not possible for the for the glucose to continue with the glycolysis and to enter citric acid cycle and then with the ATC. हमें उसमें बहुत सारे energy चाहिए, ठीक है? So कैसा होता है? So body, what the muscle cells do is they keep on doing glycolysis. जितना भी आपका ग्लूकोज है it undergoes glycolysis. जो pyruvate बना है वो directly आपका lactate बनता रहता है. Why lactate? Because this is going to give you NAD plus, and this NAD plus will be used in the glycolysis to continue to become pyruvate. अब overall net gain क्या हो रहा है मुझे यहाँ पे? More amount of ATP. Is this clear to you guys? Should I repeat once again?
Yes, ma'am.
Repeat करना है?
No, ma'am, it's clear.
Yes, ma'am.
Clear. Okay. Should I repeat? Okay, मैं repeat करती हूँ. Okay. Whatever it is, I'll repeat once again. See, normally what happens if you're breathing, walking, glycolysis होता है, आपका Cori cycle होगा, then आपका ETC होगा. You are going to get from one glucose molecule around 30 to ATP. ठीक है? So this happens in the normal condition. Now you don't have oxygen, less amount of oxygen is there, okay, and it has been done fast. So I need more amount of energy at very short interval of time. So what the muscle cells does is that it keeps on undergoing glycolysis. Okay, so जो भी pyruvate बनता है, that does not enter TCA cycle. Pyruvate से acetyl CoA बनेगा and then it will enter the TCA cycle. So this is not happening. Instead of that, pyruvate is forming lactate. Now this results in the formation of NAD plus. Now this NADA plus is required in the glycolysis to continue so that I can get more amount of ATP. Of course pyruvate मिलेगा, pyruvate lactate बनेगा और ये NADA plus बनेगा. तो NADA plus आपका glycolysis में जाएगा, so more molecules of glucose will be undergoing glycolysis and it will be giving you ATP. ठीक है? अभी clear है?
Yes, ma'am.
I guess I have got the figure, so maybe that will be also clear again. So recycling of NADH during anaerobic glycolysis. Okay, so this is what we're going to discuss. जो अभी हमने discuss किया वही चीजें same. So lactate dehydrogenase, it catalyzes the oxidation of NADH by pyruvate to give you NAD plus and lactate. ठीक है? So this NADH produced during the conversion of glyceraldehyde 3 phosphate to glycerol, glycerate, 1,3-bis phosphate is converted into NADA plus when pyruvate is converted to lactate. So this process will allow the cell to continue producing ATP under anaerobic condition as long as glucose is available. ठीक है? तो अभी यहाँ पे क्या है, मेरे को कोई टेंशन नहीं है कि मेरे पास ऑक्सीजन है या नहीं है, because my muscle cells are able to produce ATP. Now, LDH, that is lactate dehydrogenase, it works absolutely stereospecificity. यानी कि क्या? The pro-R A side. So I hope guys you know that there is R configuration and S configuration. R आपका ऐसे रहते हैं, S ऐसा रहता है. So the pro-R hydrogen at C4 of NADH is stereospecifically transferred to the pyruvate at C2 form to become L or the S lactate. Now this again generates NAD plus for the participation in the GAP-DH reaction. GAP-DH का क्या काम है? To convert glyceraldehyde 3 phosphate to glycerate 1,3-bis phosphate during the glycolysis. Okay. So this hydrate transfer to pyruvate is from the same phase of the nicotinamide ring, like that to acetaldehyde in the alcohol dehydrogenase reaction, but from the opposite phase of nicotinamide ring, like that gap in the GAP-DH reaction. ठीक है? बहुत confusion हो रहा होगा. The mechanism is not required. What you can understand over here is that you have got NAD plus and you have got the lactate. ये आपको मिलता है. ठीक है? अभी इससे पहले यहाँ पे आपके पास पायरुवेट है and यहाँ पे आपके पास NADH है. So this H is getting transferred. ठीक है? It is getting transferred to the pyruvate at the C2 form so that the pyruvate becomes the lactate. And this H, when it goes to the pyruvate, NADH becomes your NAD plus. और ये जो NAD plus है आपका glycolysis में वापस जाएगा. बस इतना समझो काफी है, because the structure and all is not required. Unless until you know the structure of NAD plus, it won't be possible for you to understand that. Okay. Now, this is really very important, guys. ये exam में आ चुका है. This is about the lactate dehydrogenase. Okay. So your lactate dehydrogenase is made up of two types of subunits, subunit A and B. कहीं कहीं पे इस A को M दिया है and then this is H. ठीक है, muscle and heart. M type subunit is predominant in the tissues which are subjected to anaerobic conditions like muscles. ठीक है, and then you have got the H type which is like predominant in the aerobic tissues like heart muscles. तो guys याद रखना, सबयूनिट A आपका M हो गया, सबयूनिट B आपका H हो गया. Okay, so LDH1 2 3 4 5. Now this is what your age, not age, sorry, page, because it's a protein, right? So polyethylamide agarogel electrophoresis. तो इसमें आप क्या देखते हैं? So which which protein is going to have more molecular weight according to this figure? Yes, Chandni?
Ma'am,
Yes, anyone?
LDH5, ma'am.
Yes, very good. So, and you need to see also, see, यहाँ पे आपका negative charge है. So, in the page, all proteins which are like subjected to the electrophoresis, they all are being charged with the negative charge. ठीक है? So, LDH5 is going to have the highest molecular weight. Good. And of course, the LDH1 is going to have the lowest. Now, in few textbooks, it will be written as see, it is B, right? So it can be written as H4. ये होगा आपका H3M या MH3 होगा, whatever ना. M2H2. Then here M3H. Then this is M4. Okay. ये कैसे आया, ये तो समझ में आया ना आपको?
Yes, ma'am.
So we have got the different 5 tetrameric isocyanates. Why tetrameric? Because 4 subunits are there. Okay. So M4, M3H, M2H2, MH3, H4, all these are there. Now, so H4 means it is for heart. It is for aerobic condition. Now M4 is for your anaerobic condition. याद रखना, ठीक है? M means for muscles, so anaerobic conditions. H is for your aerobic conditions, heart. Now, important thing, H4 LDH has low Km for pyruvate and elastically inhibited by high level of this metabolite. Whereas M4, okay, this is your M4, it has got high Km for pyruvate and it is not inhibited by it. So the other isocyanates have intermediate properties that vary with the ratio of their two types of subunits. तो आपको क्या याद रखना है? H4 है, so it has got low Km for pyruvate. M4 है, so it has got high Km for pyruvate. Okay. Now when you say high and low Km, what does it mean about its affinity towards a substrate? So yes, Shani, what can you say about it?
Low Km is high affinity, ma'am.
Yes. So low Km means आपका एंजाइम का जो सबस्ट्रेट एफिनिटी है वो आपका ज्यादा रहेगा. ठीक है? Okay. This is what I was explaining that time, like how the NADH is getting recycled during the anaerobic glycolysis. Okay. So, so you have got the over here after 3-phosphoglycerate. Okay, it is getting converted to your 1,3-bisphosphate glycerate. Now, this pyruvate is there. Pyruvate, when it becomes lactate, it is giving you NAD+. And during the glycolysis, this NADA+ is being reduced. That is, NADH बनता है जब आपका 3-phosphoglycerate becomes your 1,3-bisphosphoglycerate. So if you can remember this also, that's more than enough to explain the homolactic fermentation. Now, this is the another fate of your pyruvate. बहुत important है, कई बार आ चुका है ये. Of course, not the entire process, but they were asking like, what does this Crabtree effect means? So जब भी आपको कुछ ऐसे weird सा name लगे ना, like why it is being known as Crabtree effect or Crabtree, ऐसा क्यों बोलेंगे? Guys, can you guess why such things, such phenomenon are named like this? क्या हो सकता है इसका simple answer?
So that we can remember it easily.
नहीं, why they name it like Crabtree? कुछ भी देते
It's a
हैं.
scientist name.
हाँ, it is always like that. जब भी आपको weird सा लगे ना, just understand that it is because it has been found by that particular scientist. इसका नाम ही Crabtree है, ठीक है? Now don't ask me कि क्यों उनका नाम Crabtree है. I don't know why in the olden days people used to have such names. Anyways. So Crabtree effect. So of course Crabtree proposed about this phenomena. So he was the first person who discovered or who understood this phenomena for the first time. So he said that the Crabtree effect describes the phenomenon whereby the yeast Saccharomyces cerevisiae produces ethanol in aerobic conditions at high external glucose concentration rather than going to the TCA cycle. तो यूजुअली क्या होता है, यू हैव गॉट गुड अमाउंट ऑफ ऑक्सीजन, ठीक है, एरोबिक बोला है यहाँ पे, एरोबिक बोला है, सो यू हैव गॉट गुड अमाउंट ऑफ ऑक्सीजन, यू हैव गॉट हाई कॉन्सेंट्रेशन ऑफ ग्लूकोज, राइट, एंड देन इंस्टेड ऑफ ग्लाइकोलिसिस टू टीसीए साइकिल इट इज नॉट गोइंग. वो क्या कर रहा है, सीधा जो ग्लाइकोलिसिस में जो आपका पायरुवेट बन रहा है, ग्लाइकोलिसिस में जो आपका पायरुवेट बन रहा है वो सीधा अल्कोहल फॉर्मेशन में जा रहा है. जबकि हमने अभी क्या बोला था, अल्कोहल फॉर्मेशन कब होता है, अंडर एनारोबिक कंडीशंस में, ठीक है. तो जैसे मसल्स में आपका लैक्टेट बन जाएगा, ईस्ट में आपका अल्कोहल बनता है अंडर एनारोबिक कंडीशंस फ्रॉम योर पायरुवेट. सो दीज आर द टू फेट्स, बट इन दिस केस ईस्ट, ठीक है. सैक्रोमाइसिस सर्वेसी के केस में क्या है, when it has got high concentration of glucose, okay, very high concentration of glucose, यानी कि खाना बहुत ज्यादा है. So instead of undergoing, glycolysis to TCA cycle, it is going to produce alcohol. So the increasing concentration of glucose accelerates glycolysis, which results in the production of high amounts of ATP through substrate level phosphorylation. So what will happen? This reduces the need of oxidative phosphorylation done by the TCA cycle via ETC and therefore decreases oxygen consumption. अभी क्या हो गया है, इतना सारा आपका ATP बन गया है कि फर्दर TCA साइकिल में नहीं जाएगा. सी आपका बॉडी समझता है, राइट, आपके सेल समझते ही हैं that from one glucose it should undergo glycolysis, TCA में जाना चाहिए ताकि मुझे और एनर्जी मिले. But when the cell understands that it already have so much amount of energy कि उसको भी आगे जाने की जरूरत ही नहीं है, तो वो क्या करेगा, TCA साइकिल कंटिन्यू ही नहीं करेगा, एंड ऑफकोर्स ऑक्सीजन जो आपका ETC में यूज होना चाहिए था वो अब यूज नहीं होगा, ठीक है. अब ये क्यों ऐसा हुआ है, इट इज बिलीव्ड दैट इट इज इवॉल्व्ड एज अ कॉम्पिटिशन मैकेनिज्म ड्यू टू एंटीसेप्टिक नेचर ऑफ इथेनॉल अराउंड द टाइम व्हेन द फर्स्ट फ्रूट्स ऑन अर्थ फेल फ्रॉम द ट्रीज. ऐसा कुछ उनका बिलीव है. गाइस, ये आपको समझ में आया व्हाट इज क्रैबट्री इफेक्ट? शुड आई रिपीट?
Yes, ma'am.
समझ में आया? श्योर, बिकॉज दिस इज रियली वेरी इंपॉर्टेंट टू अंडरस्टैंड. सो कंडीशन क्या होनी चाहिए आपके क्रैबट्री इफेक्ट के लिए, दैट यू शुड हैव गुड अमाउंट ऑफ ऑक्सीजन, ठीक है, एंड वेरी हाई कॉन्सेंट्रेशन ऑफ योर ग्लूकोज, so that it undergoes your Crabtree effect. Now, why it is doing? Because after glycolysis itself, the amount of ATP is so high that the cell understands that it need not go into the TCA cycle to produce more ATP. Okay, glycolysis से ही इतना ATP मिल गया है that उसको TCA cycle से और ETC से, electron transport chain से और ATP बढ़ाने की जरूरत नहीं है. And that's why ऑक्सीजन कंज्यूम जो होता है उसकी भी जरूरत नहीं है. ओके, so this effect works by repressing respiration by the fermentation pathway dependent on the substrate. Now, for Saccharomyces cerevisiae in aerobic conditions, glucose concentration below 150 milligram per liter did not result in ethanol production. समझ रहे हो? जब आपका ग्लूकोज कॉन्सेंट्रेशन अराउंड 150 मिलीग्राम था, तब वो क्या हुआ, इट वेंट ऑन आपका ग्लाइकोलिसिस एंड टीसीए एंड ईटीसी, ठीक है. बट व्हेन द कॉन्सेंट्रेशन ऑफ ग्लूकोज वाज हाई, लाइक मोर देन 1000 मिलीग्राम, देन इट अंडरगॉन फॉर द इथेनॉल फॉर्मेशन. सो दिस organism has exhibited Crabtree effect. Now, it was the study of the tumor cells that led to the discovery of the Crabtree effect. Tumor cells have similar metabolism, which is in the tumor cells is known as Warburg effect, in which they favor glycolysis over the oxidative phosphorylation pathway. So tumor cells का क्या होता है? Like if you know guys, जब आपका ट्यूमर बनता है वो ऑक्सीजन डिपेंडेंट नहीं होता है, ठीक है, उसके पास खुद ही एनॉक्सिक कंडीशन रहता है, देयर इज लेस अमाउंट ऑफ ऑक्सीजन एंड हेंस जितना ग्लूकोज होता है वो ग्लाइकोलाइसिस करके अपना एटीपी यूज करके कीप्स ऑन डिवाइडिंग, इट डजंट अंडरगो ऑक्सीडेटिव फॉस्फोराइलेशन. ऑक्सीडेटिव फॉस्फोराइलेशन मतलब आपका एटीसी में जाना चाहिए, माइटोकॉन्ड्रिया में जाना चाहिए, वेयर द ऑक्सीजन इज बीइंग यूटिलाइज्ड. अब ये कैसे पता चला था? Like many people thought that if we provide hypoxic condition, यानी कि less amount of oxygen अगर हम देंगे तो शायद under this anoxic condition or hypoxic condition the cell will die, but tumor cells नहीं मरे. Okay, they were able to still survive. Why? Because they have got this Crabtree effect, or which is otherwise known as the Warburg effect in the tumor cells. ठीक है, so ये important चीज है समझने के लिए from the Crabtree effect. अभी क्लियर है गाइस? सेम थिंग.
Yes, ma'am.
ग्लूकोज, ग्लाइकोलाइसिस, पायरुवेट. अगर आपका इथेनॉल बन रहा है तो पायरुवेट टू डीकार्बोक्सिलेज इथेनॉल फॉर्मेशन, डीहाइड्रोजनेज विल गिव यू द नॉर्मल ग्लूकोज. यहाँ पे क्या हुआ? ग्लूकोज रिप्रेस हो गया, ठीक है, ये नहीं जा रहा है एंड इट इज अंडरगोइंग योर ग्लूकोज यहाँ पे this formation, the ethanol formation, when you have got the more amount of glucose molecules. Okay. This is the same thing like recycling of NADH during anaerobic glycolysis. So what is happening? Sugar, glucose, glycolysis, pyruvate मिला आपको, ठीक है, pyruvate decarboxylase will give you the acetaldehyde and then acetaldehyde dehydrogenase is giving you ethanol. अब यहीं से आपको एसीटेट भी मिलता है, एसिटाइल फॉस्फेट मिलता है, एसिटाइल कोए मिलता है, ये आपका नॉर्मल सिनेरियो है. अदरवाइज व्हाट इज हैपनिंग? अभी यहाँ पे एनारोबिक कंडीशन में ही आपका इथेनॉल बन जाता है और सेम चीज जो आपका पहले आपका लैक्टेट बनता था वो आपको NAD+ दे रहा था और यही NAD+ ग्लाइकोलाइसिस में हेल्प करता है टू फॉर्म NADH टू कंटिन्यू द ग्लाइकोलाइसिस, राइट? द सेम वे यहाँ पे लैक्टेट आपको NAD+ दे रहा था, अभी यहाँ पे आपको acetaldehyde to ethanol formation is giving you the NAD+. Okay, so now when glucose levels are high, yeast cells shift into the make, accumulate, consume ethanol pathway. So glucose is converted into ethanol molecules. क्यों? To get NAD+, right? Similar to your lactate. तो lactate भी किस लिए बन रहा था? क्योंकि उसका NAD+ चाहिए था. Then ethanol is released into the micro environment where it competes with the microbes. So ethanol क्या करता है? It's an alcohol, right? So it जो nearby cells रहते हैं उसको वो मार देते हैं. Excess ethanol is stored inside the cell. Once glucose is depleted, glucose repression ends. Now the cell synthesizes ADH2 enzyme, converts ethanol back into acetaldehyde. Then acetaldehyde आपका acetyl CoA बनता है, then acetyl CoA आपका TC cell बनता है. ठीक है? So guys, अभी आपको ये समझ में आ गया why it is said that this is evolutionary competitive mechanism. क्यों इसको evolutionary competitive mechanism बोला गया है? जैसे भी बोला, जैसे फर्स्ट टाइम आपका फ्रूट नीचे गिरा है ईस्ट है वहां पे, ठीक है, इतना सारा आपका फूड सोर्सेस अवेलेबल. So what happens is that if I start producing ethanol, right? So I can kill the rest of the organisms which are near to the glucose source, right? And only my species can then thrive on that particular glucose source. तो इसलिए उसको हमने competitive mechanism of evolution बोला है. इतना क्लियर है गाइस? क्यों उसको हमने उससे evolutionary significance बोला था? Now it is clear?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay, so now we'll just see like the energy yield of the aerobic versus anaerobic glycolysis. So even though I have not taken the entire thing कि glycolysis और TCA cycle and ATC के बाद हमें मिलना चाहिए, because right now I'm just talking about the glycolysis, right? So still जब हम आगे जाके compare करेंगे तो हमें पता चलेगा. So in the case of aerobic and anaerobic glycolysis, each molecule of glucose generates 2 molecules of ATP. ये तो हमें पता है, ठीक है? So from glucose because of the glycolysis we get 2 ATP molecules. ठीक है, 2 molecules of NADH and 2 molecules of pyruvate. Okay, and हमें पता है NADH is equivalent to 2.5 ATP, then 2 molecules of pyruvate. The net energy yield in anaerobic glycolysis is 2 molecules of ATP per molecule of glucose as NADH is recycled to NAD plus by reducing pyruvate to lactate. तो यहाँ पे आपका ये यूज हो जा रहा है. Now, whenever oxygen is available, the cytosolic NADH can be oxidized via shuttle system from mitochondria, and pyruvate can also enter into the mitochondria. तब आपका TC cycle में वो enter करेगा. ठीक है. So the NADH gives you around 2.5 ATP molecule if oxidized. अगर वो 1.5 देगा if it is oxidized by the glycerol 3 phosphate shuttle. So both these shuttles we'll talk about. Now the 2 NADH molecules produced during glycolysis can lead to 3 to 5 molecules of ATP depending on the shuttles, and because each pyruvate produced can give rise to 12.5 molecules of ATP. Altogether हमें aerobic glycolysis में कितना मिलेगा? Around 30 to 32 molecules of ATP. Right? So कौन सा better हुआ? Aerobic, right? Because anaerobic में क्या हो रहा है? आपको कितना मिल रहा है? 2 प्लस पायरुवेट से कितना मिलेगा? अराउंड 12.5. हाँ, तो आपको कम ही मिल रहा है, राइट? तो अगर मैं पूरा ऑक्सीडाइज कर रही हूँ इन द प्रेजेंस ऑफ ऑक्सीजन, the entire glycolysis CC cycle etc is happening, then I'm getting more amount of ATP. So of course aerobic is better than the anaerobic glycolysis. Okay, now tissue dependent on the anaerobic glycolysis. So many tissues includes like red and white WBCs, kidney, medulla, the tissues of the eye and skeletal muscles. They rely on anaerobic glycolysis for at least a portion of their ATP requirements. Now, why do you think it is because of the anaerobic glycolysis? क्यों उनको rely करना पड़ता है? Why? Because they are in continuous use. ठीक है? So there are chances that आपके पास oxygen नहीं मिल रहा हो, but they need to function their job. They need to do their job. So they are dependent on the anaerobic glycolysis also for their ATP requirement. Okay. Now, अगर आपके पास lactic acid generate हो गया because of the anaerobic glycolysis, so it will be secreted out from the skin in the form of the sweat, and that sweat is going to have that antibacterial agent. Okay. So many large tumors use anaerobic glycolysis for ATP production and lack capillaries in their pores. So guys, यहाँ पे हम खत्म करते हैं about the glycolysis. अभी तक का clear है? सबको समझ में आया जो भी हमने आज discuss किया?
Yes, ma'am.
Yeah. Okay. I'll be sharing this PPT today itself so that you can go through again. And अगर कोई doubt होगा तो you can ask me in the next lecture. Okay. Okay. Now it's not over, okay? So we have got one more important thing that is known as Cori cycle. Again, of course, by the person named Cori. So in the liver, the pyruvate is used to synthesize glucose. यानी कि gluconeogenesis. You are going to synthesize glucose. अभी हम ये क्या कर रहे थे? Glycolysis कर रहे हैं, यानी कि हम break कर रहे हैं glucose को, ठीक है? So which is returned to the blood. The cycling of the lactate and glucose between the peripheral tissues and liver is called the Cori cycle. अभी हम Cori cycle क्यों डिस्कस करें? Because right now we discuss about the glycolysis. आगे जाके अभी हमें gluconeogenesis सीखना है, ठीक है? From the next lecture onwards, so we need to talk about the gluconeogenesis. So कैसे glycolysis and gluconeogenesis connected है? That is your Cori cycle. So see, यहाँ से हम शुरू करते हैं from the glucose का glycolysis से. So you have got the glucose. ठीक है, ये भी कोई भी आपका respiring tissue है. Okay, so glycolysis हुआ, आपको यहाँ पे lactate मिल गया, 2 ATP molecules मिल गए. The lactate will be transferred to the liver. From the liver, this lactate is going to convert it into the pyruvate, and फिर वहाँ से आपको gluconeogenesis मिलेगा, and that is glucose. So ये glucose वापस आपके blood में जाएगा और फिर blood से फिर respiring tissues में जाएगा. इतना क्लियर है? ये बहुत ही, it is just the outline of the Cori cycle. Later we are going to discuss in detail the Cori cycle. ठीक है? अभी देखो ग्लाइकोलिसिस के यहाँ पे 10 स्टेप्स हो गए आपके. ओके, सेम वे ग्लूकोनियोजेनेसिस के रिवर्स 10 स्टेप्स होंगे. ये थोड़ा सिंपल ही रहेगा. ओनली डिफरेंस व्हाट यू सी विल बी इन दोस 3 इरिवर्सिबल रिएक्शंस. ठीक है, तो वो ही 3 जो स्टेप्स होंगे वही यहाँ पे वो डिफर करेंगे इन द नेम ऑफ द एंजाइम्स. रेस्ट ऑल इट इज गोइंग टू बी द सेम. एंड व्हाट वी नीड टू रिमेंबर इज the lactate, how the lactate is being transported from the tissue through the blood to the liver. ठीक है, ये और हमें एक शटल है उसके बारे में पढ़ना होगा. So that is the connecting link between your glycolysis and gluconeogenesis. Okay, so this outline is clear to you guys आज के लिए?
Yes, ma'am.
Okay. So glucose is an indispensable metabolite. So of course the brain requires at least 50% of its calories in the form of glucose. RBC भी glucose पे dependent है. Glucose is precursor of other sugars needed for the synthesis of nucleotides, glycoproteins, glycolypids, etc. It is also needed for NADPH, which is the supplies reducing power for the biosynthesis and detoxification. So in short, glucose के बिना we cannot survive. Okay. So yeah. So that's all for today. And यहाँ पे आपका glycolysis खत्म होता है, ठीक है? So from tomorrow onwards we'll be doing the gluconeogenesis. And once this glycolysis, gluconeogenesis आपको खत्म होता है, then we can talk about the citric acid cycle and electron transport chain. Okay, that will finish or that will cover the entire your carbohydrate metabolism. Okay, so now test yourself. So this is from the PYQ. The question is, the following are the statements about pyruvate kinase. So what is the function of pyruvate kinase? Yes, क्या है? ग्लाइकोलिसिस में पायरुवेट काइनेज का क्या काम है? So if you remember your glycolysis, so pyruvate kinase का क्या काम रहता है? To convert your phosphoenol pyruvate to pyruvate. आपका जो लास्ट स्टेप है, राइट? दिस इज योर लास्ट स्टेप, स्टेप 10. तो यहाँ पे आपका पायरुवेट काइनेज आता है. ओके, सो पायरुवेट काइनेज है, फॉस्फोइनोल पायरुवेट से आपका पायरुवेट बन रहा है. सो रिमेंबर टुडे इन द स्टार्टिंग ऑफ दिस लेक्चर आई सेड दैट द हाई एनर्जी फंक्शनल ग्रुप व्हिच हैव गॉट द मैक्सिमम एनर्जी, इट इज द इनॉल ग्रुप, राइट? सो इट कैन गिव यू ADP से आपका ATP बनाता है. So this is a step at which you actually synthesize ATP. Okay, अब question क्या है देखते हैं. So ATP is an allosteric inhibitor of PK. Is it true? Is this statement is true? Fructose 1,6-bisphosphate is an activator of PK. ADP is an allosteric inhibitor of PK. Alanine is an allosteric modulator of PK. Yes, guys. क्या लगता है?
What is the concept of that allosteric inhibitor?
Allosteric मतलब सपोज ये आपका PK है, ठीक है, पायरुवेट काइनेज है, यहाँ पे सब्स्ट्रेट बाइंड करना चाहिए, इनहिबिटर आपका यहाँ बाइंड करेगा एंड अफेक्टिंग द रेट ऑफ द एंजाइम एक्टिविटी. अभी क्लियर है? So ATP is an allosteric inhibitor of PK. So ATP inhibits PK activity. Now, is it true?
No, ma'am.
Why? This statement
Because here ADP is converting to ma'am. How it?
हाँ, see, ATP is allosteric inhibitor of PK. So what does it mean? PK is synthesizing your ATP. अब ATP का concentration high हो जाएगा. So what will happen? This will go and bind over here and inhibits the activity of PK, saying that बस और मत बनाओ, है ना?
Yes, ma'am.
Okay, so
Yes.
if I have got more concentration of ATP, I can inhibit the activity of PK, and that is your allosteric inhibitor. So this statement is true. अब यहाँ पे देखो, ADP is an allosteric inhibitor of PK. अब ये statement गलत है. Why? Because PK is converting ADP to ATP. Now getting the point, Chandni?
Yes,
Yes,
ma'am.
ma'am.
पक्का clear है ना? See, once again I can tell. So you have got your PK over here, pyruvate kinase. इसका काम क्या है? To convert phosphoenol pyruvate to pyruvate. ठीक है? अब इसी प्रोसेस में it is also converting your ADP to ATP. बट अगर एटीपी कॉन्सेंट्रेशन अभी ज्यादा हो गया है, सो व्हाट विल हैपन, इट कैन कम बैक एंड बाइंड टू इट्स एक्टिव साइड, एलिस्टरिक साइड, अनदर साइड, बिकॉज एक्टिव साइड में तो आपका फॉस्फेट पाइरुवेट एंड वो सब है, दूसरे साइड में आके बाइंड करेगा, कंफर्मेशन चेंज होगा पीके का, दैट इज inhibiting the activity of PK. Why? It's stop now. We don't want more ATP synthesis now. We have got enough in the cell. So stopping the activity of pyruvate kinase. Okay. So that is your allosteric inhibitor. But now in the statement C, ADP is an allosteric inhibitor बोला. अब ये गलत है भाई, because ADP से तो वो ATP आपको देता है. So if you have got more concentration of ADP, see, if you have got more concentration of ADP, what does it mean? It means the cell has less amount of ATP and more amount of ADP. So your pyruvate kinase will convert into ATP then. So वो inhibitor नहीं हुआ, activator हुआ. ठीक है? So the statement is false. A सही है. Okay. So what all options are left now? So either it can be 1, 2 और 4. है ना, तो हमने यहाँ पे थर्ड स्टेटमेंट जो है वो हमने कैंसिल कर दिया. ओके. now fructose 1,6 bisphosphate is an activator of PK. yes, tell me. so इसलिए मैंने बोला था आपको मालूम होना चाहिए.
Yes, ma'am, it is true.
It is true. Very good. So if you remember the glycolysis step, then you can talk about, then you can tell all this. So अभी हमें मालूम है A सही है, B सही है. So this option also gone. अभी हमारे पास क्या है, 1 और 2. Now what? No, 1 भी गया because C हमने already cancel कर दिया ना. Okay, so option is A, B and D. What is the last? Alanine is the allosteric modulator of PK. So this will study later when we talk about the regulation. So anyways, आपको फिर भी आंसर मिल गया. Option B is the correct answer. ठीक है. So अगर आपको 4 में से 3 options के बारे में भी पता है, more than enough. Okay. अभी सेम वे एक नोट की तरह बना लो इसको. लेट मी जस्ट राइट इट फॉर यू. सी, ये हो गया पीके, पायरुवेट काइनेज, प्लस एंड माइनस. तो प्लस मतलब एक्टिवेटर, माइनस मतलब इनहिबिटर. सो प्लस मतलब ए बी पी इज गोइंग टू एक्टिवेट पायरुवेट काइनेज का एक्टिविटी. एग्री? अगर एडीपी की जगह एएमपी भी लिखा होता तब भी सही होता है. हाँ, क्योंकि AMP क्या है, अगेन इट मींस द सेल हैज गॉट लेस अमाउंट ऑफ ATP. करेक्ट? सो बोथ द सिनेरियो इट इज गोइंग टू एक्टिवेट PK का एक्टिविटी. एंड ऑलरेडी एज वी सेड फ्रक्टोज 1,6 बिस्फॉस्फेट is also going to activate the activity of PK. Now what is going to inhibit the activity of PK? ATP, as just now we have discussed, because more amount of ATP, it means cell does not need more ATP. So stop now. Then if the cell has got more amount of acetyl CoA. Now why? Because acetyl CoA अगर ज्यादा है, it means the cell is already having so much amount of energy. acetyl CoA ज्यादा हो जाएगा तो यानी कि मुझे और ग्लाइकोलाइसिस करने की जरूरत नहीं है. acetyl CoA भी आपका TCA साइकिल में जाएगा to give you more amount of ATP. So if you have got more amount of acetyl CoA, so I don't need more further glycolysis to happen. So it is a negative impact on the PK. Clear, guys?
Yes, ma'am.
Sure, clear है ना? HCl CoA का समझ में आया?
Yes, ma'am. Yes, ma'am.
So again I'll say, see, from glucose, glycolysis, pyruvate, acetyl CoA हमें मिलता है. ठीक है? acetyl CoA आपका माइटोकॉन्ड्रिया में जाके TCA साइकिल बनाता है. ठीक है? अभी मैंने बोला acetyl CoA बहुत ज्यादा है हमारे पास. We have got lots of amount of the concentration of acetyl CoA is very high. So it means it should actually stop your glycolysis further. ठीक है? So it means it is the negative impact on the PK. Okay? अभी clear है ना सब कुछ? Now from this we can also say that alanine is also coming in the minus half. Okay, so that we'll study later when we talk about further pathways. Okay, so now you know the answer. Okay, so that's all today, guys. Thank you for listening. और कोई doubt है आज की classes में?
No.
Okay.